Welcome to this tips and tricks video for the ibidi microslide I Luer 3D. Carefully remove the protective foil with a scalpel, ensuring the sticky tape underneath stays in place. Use tweezers to fully detach the foil. Next, fill each well with exactly 16 microliters of gel. It can help to hold the pipette at a 90-degree angle during pipetting, so the gel evenly spreads across the entire well. Do not shake or tilt the slide after adding the unpolymerized gel to keep it in place. For this video, we placed scale paper beneath the slide elevated on a rack. The scale paper helps verify whether the gel surface in the wells is flat or if the volume is too low or too high. The first well contains exactly 16 microliters, producing a flat surface without magnification effects. In the second and third well, we purposefully added too little or too much gel. Observe the demagnification or magnification effect on the scale paper when viewed from above and adjust if needed. After filling all 3 wells with gel, place the slide on a flat, stable surface, such as a petri dish on the bench. A stable support prevents the slide from bending during coverslip sealing, and the petri dish protects the slide from scratches. Remove the protective foil from the supplied coverslip using the blue tab, then center the coverslip on the sticky surface. Avoid pressing directly on the channel area. This can cause the flexible coverslip to touch the unpolymerized gel, leading to gel adhesion and potential channel blockage. Press down firmly with your fingers or the back end of tweezers to eliminate all air bubbles. Air bubbles appear as lighter areas, while darker regions indicate a proper seal. Ensure the coverslip is completely sealed to prevent leakage later. Cover the Luer adapters with the supplied caps to maintain sterility. Let the gel polymerize under appropriate conditions, for example the incubator. Since hydrogels can swell or shrink during polymerization, check the surface afterward and readjust accordingly. If the gel detaches from the well edges after polymerization or later, pre-coat the well with the same matrix. This creates an adhesive layer that helps secure the gel and prevents detachment. The hydrophilic ibidi coverslip allows hydrogels to spread a bit more easily compared to the hydrophobic untreated version. If your pipette setting does not achieve meniscus-free filling, adjust the volume slightly and confirm using the scale paper. Remove caps on top of the Luer ports and use a syringe to inject cell suspension into the channel. Avoid using a pipette as this increases the risk of introducing air bubbles. Carefully insert the syringe into one of the Luer ports and twist until it is securely sealed. Slowly inject about 250 microliters of cell suspension, ensuring the channel fills evenly without trapping air bubbles along the edges. Completely fill the opposite Luer port as well. Otherwise, removing the syringe may draw an air bubble into the channel as shown here. If this happens, reinsert the syringe fully into the port and add more cell suspension. Remove any remaining cell suspension from the Luer ports with a pipette so cells only grow in the channel. Take care not to remove the cell suspension from the channel. Cover the Luer ports with caps, place the slide in a petri dish and allow cells to adhere in the incubator. Once cells have fully adhered, you can either add 60 microliters of medium to each Luer port for static cultivation or connect the slide to flow. Clamp the perfusion set tubes to avoid medium spillage. Overfill each Luer port with medium until you see an upright meniscus or medium bump. Pull one elbow Luer downward so any air trap can escape upward. Slowly push and twist the elbow Luer into the Luer port until you feel resistance, ensuring a leak-free connection. Do not worry about medium spillage at this point. This is necessary to avoid introducing air bubbles. Repeat this for the second elbow Luer. Remove all spilled medium with a sterile tissue and release the clamp. Now you can start your flow experiment. If cells detach under flow or are not 90 to 100% confluent, we recommend continuing static cultivation until they reach full confluence before applying flow.